Our next presentation is Aspen Neurosciences, and I'd like to welcome Kim Ranieri, Chief Technology Officer, to the stage.
All right, thank you. I'd like to thank the organizers for having me. My name is Kim Ranieri. I'm the CTO for Aspen Neuroscience. We are an autologous iPSC company. I have a safe harbor statement. I might make some forward-looking statements as we go through this presentation. But to get us started, we've made some really great progress over the last year. I'm really excited today to be up here to share some of that with you. As I mentioned, we're an autologous iPSC platform company. Our lead program is in Parkinson's disease, where we've got fast track designation. We've dosed our first cohort of patients in our phase 1/2a clinical trial and have established surgical safety data on that. I'm going to show that in a little bit. We have data readouts that will be coming in early 2025, and we have used digital health technologies as part of that as some exploratory endpoints, and we're really excited to see some of that data that's coming along with that. On the manufacturing side, we've made really good progress. We announced a new manufacturing site last week, as well as good progress on automating our manufacturing process in a collaboration with a company called Cellex Technologies, which I'm going to go into a little bit more. We really believe our science and our approach has already been validated, and there's a lot of activity and a long history in the space of cell replacement therapies for Parkinson's disease. And so really many have come before us in this space, and we're learning from their experiences, and I like to think we're, to some degree, standing on the shoulders of giants here when we're trying to move our therapy through the clinic. And we really believe that if you can get the right cells to the right place, that this is going to be a transformational therapy. One of the kind of key distinctions of our therapy and our approach is that it doesn't require immune suppression, and it's the use of immune suppression has really a high burden on these types of patients, increasing risk for infection, as well as other adverse events. We have a pipeline. Our pipeline is focused on Parkinson's disease, but we do have some other CNS indications. And importantly, though, our pipeline really can be leveraged across a host of cell types, not just CNS, but beyond. And if you're interested in partnering with us, come see us. Our vision really is to be, the leading autologous iPSC company, and we're planning to do that by leveraging our deep knowledge and experience in stem cell biology, as well as our bioinformatic platform, as well as our in-house CMC capabilities. Our bioinformatics platform really allows us to choose the best cells for the patient, and this really starts with our pluri-test, where we ensure that we have the highest quality iPSCs. And in the case of Parkinson's, we then differentiate those into dopamine precursor cells. And on the drug product side, we have a suite of RNA-seq-based assays like NuriTest for ensuring the proper identity and maturity of those dopaminergic precursors, as well as assays that can predict engraftment and dopamine release potential. And so we do these bioinformatic assays for every batch, ensuring that we have the highest quality cells for each patient. So a little bit about our phase 1/2a trial. It's safety, tolerability, and dose response. We have 2 dose cohorts. As I mentioned, we've dosed the first cohort, and we're well into dosing our second cohort. We've already completed all the manufacturing for this study, and we believe we'll complete the cohort 2 before the end of the year. We're going to have multiple readouts for both primary and secondary endpoints, some of which should give us an indication of biological effect. And we're planning to use this trial-ready cohort as we look forward into our phase 2B, so we can kind of prime that pipeline and get patients basically in the funnel for the next clinical trial. So I'm going to show you now some data that was shared by our lead neurosurgeon, Dr. Paul Larson, at the movement disorder meeting a few weeks ago. This shows the surgical technique where we use a bilateral approach, doing a occipital burr hole on each side. We do 2 tracks, a dorsal and a ventral track. We utilize a clear point cannula system with a proprietary delivery device, which we call the Aspen metered delivery syringe. If you can see, let me go back. Here we go. See here, the cannula goes in, and then we retract that cannula approximately 2 millimeters and leave cell deposits across the posterior putamen. The data that we have is from the first 3 patients. We've got over 3-month follow-up with good safety data. There's been no surgical serious adverse events. And as you can see with the visualization of the cell deposits, we can see those during the surgical procedure so that we know we're getting the right cells to the right place. In conclusion with this, we've shown that the initial low dose can be safely administered, and we're now testing the same approach with the high dose. So moving along to some of the manufacturing activities. As I mentioned, we announced our new 22,000 square foot manufacturing facility in Torrey Pines, San Diego. This is the facility we use to manufacture the product for our phase 1/2A. We're actually continuing to manufacture some additional product for additional cohorts. This particular facility has about 2,000 square foot of clean rooms, as well as another 8,000 square foot of expansion space. And we're planning to use that as we look forward to our later clinical trials, and for that space to be utilized to build out our automation platforms. At Aspen, we've got a pretty deep experience in iPSC generation. We've made over 500 cell lines across the different groups. And of those, we've made 100 different GMP cell lines from the various patients that we've manufactured for. What we're working on now is a thaw and inject formulation for our dopaminergic product, so that we can eliminate any dose preparation prior to the surgical procedure, as well as working on automation platforms, which I'm going to tell you a little bit about right now. So we announced a collaboration and licensing agreement with CellX Technologies, and we're going to work with them to further develop their Celligent platform for GMP manufacturing. This platform really combines state-of-the-art robotics, liquid handling, imaging and analysis, all encapsulated inside an environmental chamber, along with automated incubation, with computer-driven algorithms where we can deploy AI and ML for automated and what we believe in the future autonomous self-driving manufacturing operations. And we believe this platform has the potential to automate over 70% of our manufacturing process. We'll primarily use it to address iPSC generation and scale up of the iPSCs, leading into the differentiation process. So we've been working with this platform for over a year, first evaluating it and now really diving into developing the process of fully automated iPSC manufacturing. We've executed thousands of unit operations across multiple Parkinson's disease cell lines and have produced high-quality cells comparable to our manual process. The really nice thing about this particular platform is that it's not a huge leap from our manual process, and it really kind of simulates a lot of the manual operations that take place by the human operators. The specific unit you can see here is this is inside the environmental chamber. The black unit here is the core imaging and deck where the media handling and liquid handling occurs. You've got a robotic arm that takes the plates from the automated incubator tray and moves them onto the deck and delids it. And the really nice feature about the system is that it can do both positive and negative cell selection. Again, similar to what human operators do. And we've been able to roll that out and employ that in our automated process. And actually what we found is that selection process is actually more precise than what the humans can do. So we really anticipate that this platform will be able to handle hundreds of patients in parallel on an annual basis. And so really now where we're kind of diving in is developing these algorithms where we can then deploy the AI and ML in the manufacturing process. And a good example of this is this image segmentation algorithm that we like to call Aspen Vision. And on what you can see here is in this blue is the iPSCs. The red is the differentiated, the yellow is kind of the unknown class, probably the iPSCs moving into the differentiated state. And then what we can do is we can have the device either pick the cells that we want or remove the cells that we don't want. And it can also be used for things like confluency measurements or decisions on whether to feed or passage the cell culture or rank the cell cultures from a quality perspective and make kind of go, no-go decisions with moving that forward or eliminating it. And really one of the key things as you think about developing these algorithms is that you need good data sets, right? So we've had a really focused effort over the last year to develop a very robust data set to train the algorithms such that in the future we'll be able to run this autonomously, and that's how we feel like we'll really be able to bridge the gap between the current manual processes in iPSC generation to an automated process. So with that, I'll kind of wrap up. As I mentioned, we've completed the cohort 1 with good safety data. We're on track for dosing the remaining patients. We're going to have some clinical readouts early next year. We've got our new manufacturing facility online, and we're making some really nice progress on the automation of our manufacturing process. So I'd like to acknowledge the patients who have signed up for the clinical trials. It's not anything insignificant to raise your hand and get brain surgery on experimental treatment along with their caregivers, as well as a lot of the institutions that have supported us to bring forward this therapy. Thank you very much.